17.3, muscle tissue and the sliding filament mechanism. This is a diagram of an individual muscle cell, also called a muscle fiber, because skeletal muscle cells are very, very long cells. The plasma membrane of the muscle fiber is called a sarcolemma, which is just a fancy name for the plasma membrane. Inside that sarcolemma are long bundles called myofibrils, and then each of these myofibrils contains filaments. The filaments are made of the contractile proteins. Myosin makes a thick filament, and actin makes the thin filament. The T-tubules are some invaginations off of the sarcolemma. So those T-tubules you can see in the diagram are continuous with the sarcolemma, and they turn and penetrate deep down into the cell. Another structure we need to identify is the sarcoplasmic reticulum. This is the ER, the endoplasmic reticulum of a muscle cell. Its primary function is to store calcium, which is going to be very important as the final trigger, which initiates contraction. The myofibril that contains the filaments, myosin and actin, again these are the thick and the thin filaments, and they are arranged in a very specific structure, the sarcomere. A sarcomere is the contractile unit of muscle. We need to see the structure of the sarcomere. So here in this diagram, this represents an individual sarcomere. The boundary of the sarcomere is the Z-line, and then extending from the Z-line in towards the center of the sarcomere are the actin or the thin filaments, and then spanning the midline are the myosin thick filaments. So when contraction occurs, we'll look at the mechanism in just a moment, there are some heads on the myosin, and they're going to reach up and grab the actin, and they pivot. And whenever this happens, it's going to shorten the sarcomere and contract muscle. There are also some regulatory proteins that we'll identify, and we'll see these on the diagram. Troponin and tropomyosin are some regulatory proteins that are also associated with the sarcomere. So let's look at the mechanism of muscle contraction. This is called the sliding filament mechanism. And so we get a nerve impulse arrives on a motor neuron. So that action potential arrives at a place called the neuromuscular junction. So we have a synapse that takes place. That motor neuron releases the neurotransmitter acetylcholine. That acetylcholine binds to receptors on the postsynaptic membrane, in this case this muscle cell, and that's going to open some channels, some calcium channels on the sarcolemma, and so we can begin another action potential in the muscle sarcolemma. So very similar to some of these events that we saw back in lesson 15 when we were discussing synaptic communication. So you see the axon bulb of the motor neuron, the little synaptic cleft, the sarcolemma, the acetylcholine is released. It's going to bind to some receptors, opens some ion channels in the sarcolemma, and now we have an action potential moving across the sarcolemma. The action potential is going to spread across the sarcolemma, but then it also turns and goes down those T-tubules. So the action potential penetrates deep down into this muscle fiber by moving down the T-tubules, and on either side of the T-tubule is the sarcoplasmic reticulum. The excitation of the T-tubule causes the sarcoplasmic reticulum to release calcium. So then calcium is going to be able to diffuse throughout this muscle fiber, and the calcium is going to bind to those regulatory proteins of the sarcomere. Whenever this happens, there's some conformational change in these regulatory proteins, and it uncovers a binding site on actin, the thin filament. Anytime this binding site is uncovered on actin, the myosin head is going to bind to the actin, and then it will spontaneously pivot to contract the sarcomere. When the myosin head actually binds to the actin, this is called a cross bridge formation. We'll see a diagram that illustrates this in just a moment. So beginning up in the upper left-hand corner, you see the actin of the thin filament. The little dark dots represent the binding site. In relaxed muscle, the regulatory protein tropomyosin covers those binding sites. However, if this muscle is stimulated, calcium binds to the troponin, and the troponin moves the tropomyosin, and this is what uncovers those binding sites on the little actin monomers. Again, anytime those are uncovered, now move around towards the lower right-hand corner, and this shows our cross bridge formation. Anytime those actin binding sites are uncovered, the myosin head will form the cross bridge, and then it will spontaneously pivot. That pulls on the thin filament. That thin filament is connected to the Z-line on either side of the sarcomere, and so the entire sarcomere will contract. The energy needed for contraction does come from ATP, and actually the ATP is needed to cause the myosin to let go of the actin. After it is pivoted, an ATP molecule is necessary. That causes detachment. We break the cross-bridge formations, and then that also recocks the myosin head. If the actin binding sites are still uncovered, then we're going to repeat the cycle. And so the little myosin heads keep forming the cross bridge, and then we pivot to contract. So here is the diagram of the sarcomere we saw just a moment ago. You can see the heads extending up off of the thick filament of myosin, and the heads on the right-hand side are going to grab and pivot to the left, which pulls that right Z-line. The myosin heads on the left-hand side are going to pivot towards the right, and that pulls the left-hand Z-line. So this entire sarcomere will contract. So here's my first ever home movie. So again, we have the Z-lines. You see the actin thin filaments extending from the Z-line towards the middle of the sarcomere. The large orange bands would be the thick myosin, thick filaments. And so if cross bridges form, the myosin head grabs the actin and pivots, and so the entire sarcomere shortens. This is called the sliding filament mechanism, because these thick and thin filaments slide past one another. Now notice, neither the thin filaments nor the thick filaments have gotten shorter. They have simply slid past one another. That pulls the I band, which is the boundary of the sarcomere, shortening the sarcomere, and will shorten the muscle.